everyone. Welcome to the second episode in YouthConnect's Research and Healthcare Interview Series. My name is Joy. I'm a first-year med-sci student at Western University, and I'm also Scientific Director at YouthConnect.
Hello, everyone. My name is Rashi, and I'm a second-year undergraduate student. I'm pursuing my triple majors in biotechnology, biochemistry, and genetics, and I'm also a Scientific Director at YouthConnect.
Hi everyone, my name is Fatima and I'm a grade 12 student in Ontario, and I'm also a scientific director at YouthConnect.
Right, and just a brief overview about what YouthConnect is. YouthConnect was founded in late 2021 as a scientific outreach initiative for high school students, and we focus on providing resources to students with an interest in the field of healthcare or research by posing as an outlet for high school students to learn more about research that is currently occurring in a variety of fields and from experts in those fields. And so without further ado, please join us in welcoming Dr. Seguin from the Department of Physiology and Pharmacology at Western University, where she's the principal investigator of the Seguin Lab. And Dr. Seguin conducts research relating to the function of the spine and intervertebral discs. Dr. Seguin, would you like to introduce yourself as well?
Sure. So I'm Cheryle A. I am a professor now at the University of Western Ontario, where I've been for the last 13 years now, and have a lot of fun running a basic science research program in the skeletal biology research group there.
And that's amazing. Thank you so much for your introduction. Before we get into the research presentation, we would like to ask you a quick round of questions to get to know you better. So the first question is, what's one piece of lab equipment that you can't live without?
Oh goodness, probably the real-time PCR machine or a microscope. Hang on, that's a tough one.
Well, great answers. Another question is, what is one quality that makes a good researcher?
Curiosity. Absolutely.
That's right. So what is one piece of advice that you would like to give to high school students who aspire to enter healthcare or research?
Great question. Take every opportunity you have to just try something new. Often we think we know what we want to do, and we think, there's one path in life. And until you try things, you don't know how much fun they might be.
Well, thank you for that advice. And as I'm in high school, I will definitely be taking that in. So thank you so much for answering those quick questions. So now we can move into Dr. Seguin's presentation, where she will introduce us to her work. So take it away.
So today I'm super excited to take this opportunity to talk to you about the basic research that my lab does, trying to understand this really big problem of back pain, and why perhaps to maybe interest some of you to better understand what basic research actually is and how it fits into our understanding of biology, and hopefully to finding solutions to really important problems. So I was asked to start with a little bit about myself. And so I put together one slide to tell you where I came from and what I'm doing. So I grew up in a rather small city in northern Ontario, Sault Ste. Marie, which is known for mining and forestry and lakes and mountains, and not so much for basic research. I grew up speaking French as my first language, surrounded by really neat experiences and a different lifestyle up there. But when I was in high school, I myself got sick with rheumatoid arthritis and really started thinking about what diseases were and how they worked. And that really got me interested in this area of biomedical sciences. And so I actually did my undergrad at the University of Western Ontario a lot of years ago, where I did my first degree in genetics, and then stayed at Western for a few years to do my master's, working in the area of cartilage cell biology. So I worked with a wonderful mentor, Dr. Suzanne Bernier, in the Department of Anatomy, trying to understand how cells sense disease environments and what sort of signals change their biology in the context of disease, and had a really lovely time understanding kind of fundamentally how cells worked. From there, I decided that I was up for a challenge and really wanted to move from, really small cells stuck in dishes to bigger, to the next scale up, which usually is tissues and growing things. So I went on to the University of Toronto, where I did my PhD in tissue engineering with another wonderful mentor, Dr. Rita Kandel, where I made the change from working in the area of cartilage biology to working in the area of spine and intervertebral disc, and had some really great experiences. And this is for the first time working in teams with people with totally different expertise. So as a young PhD student, I was working with engineers, making cool materials that I was putting cells on, and we were working in big animal models. I got to scrub into a cow surgery, trying to implant the tissues that I'd grown. So these really neat experiences that, for me, really solidified the value of basic research. That I was always intrigued with trying to fix problems and trying to understand how things work and take them apart and then fix them again. And for me, instead of the field of medicine, the field of biomedical research really provided those opportunities, because you could understand things at this like super basic level and then hopefully be able to apply those to fixing a problem. And so it's my plug for, an interest in medicine and in disease doesn't necessarily have to be a path in clinical sciences, that there's a lot of great value to understanding science. I then had a really phenomenal opportunity to do a postdoc, which is, what you do between a permanent career in academia and being a trainee. I was at the hospital for sick kids, and I was lucky enough to be in one of the first labs working in human embryonic stem cells. When I was a tissue engineer, I understood that if we had really healthy tissue, we could grow really healthy tissue, but that's not a really great real life application, because we don't always have a source of really healthy tissue to make products with. And so I decided that the field of developmental biology and understanding stem cells and where they came from and what you could do with them was really exciting to me. So I spent a few years there until I was recruited to back to Western in 2009 as an associate professor, where I was able to establish the Seguin lab and take all of those things that I had learned from all of these different experiences, life experiences and academic experiences, and for the first time be able to sit back and just think about what questions we wanted to address. So I'll tell you a bit about the big questions that we like to address in my lab. And when we talk to students, when we talk to funders, the agencies that fund our basic research, the argument is pretty easy to make, because low back pain is the most frequent musculoskeletal problem in adults. It's the second most common reason people go to the emergency room. And in Canadians, about 84% of people will experience severe back pain at least once in their life. And close to 40% of our population will actually experience chronic back pain. So it's a really big problem. And the reason that it's so compelling to work in this area is that there is nothing we can do about it. So besides managing pain, there are really no interventions to slow the diseases that are causing the pain. And for my lab, we focus on understanding what goes on. And I've thrown this one up here. There's these massive clinical studies called the Global Burden of Disease Studies that map out the biggest reasons for disability across the globe. And not surprisingly, the most recent one identified back pain as the most common cause for disability worldwide. So we don't have to stretch very far to say why what we do is really important. And we tackle the area of back pain by studying the joints within the spine called the intervertebral discs. So for us, this is really neat. Not all back pain is associated with disc problems. If it was, we probably would have figured it out by now. There's lots of things that contribute together to cause chronic pain, but in about 40% of those cases, there is a structural problem in the joints of the spine. And so this is the tissue that our lab studies. We think that understanding what goes wrong in the spine associated with pain would be really important to trying to come up with ways to fix it. Joints in the spine are really intriguing. They're quite complex. There's all these different tissue types that have to work together like a team to be able to fulfill the function of the spine, which is to absorb mechanical load from us walking around or running every day, and enable flexibility. Because if you didn't have joints along your spine would be a solid rod and we wouldn't move very well. And so much of our posture, so much of our activity depends on the function of our spine. And so for us, these are really intriguing questions. And what we really focus on is understanding the difference between a healthy disc and a diseased disc, so that if we can understand what goes wrong, maybe we can intervene or try to fix it. And so I'm going to tell you 3 really abridged stories about what we do. And I've picked these 3 stories because I think they give you a different view of some of the tools we use, maybe some of the questions you can use, or questions you can ask in basic research, just to kind of give you a flavor of how we approach questions and maybe some of the neat things that we can do. So the very first thing that we asked was, where do cells come from that make up the intervertebral disc? And you might be thinking, that seems weird. We should probably know that by now. And it's really quite surprising that our understanding of the biology of the spine kind of lags a lot of our other bone and joint tissues in terms of our knowledge. And back in 2009, when I started my lab, we really didn't know the answer to this. And so the intriguing part and the tissue that we're focused on is this middle of the disc, the nucleus pulposus. And it's kind of like the jelly in a jelly donut. Its job is to absorb a bunch of water and enable this disc structure to resist compression, kind of like the water in a water balloon. If you push down on it, it can't go anywhere because it's confined. And this tissue is made up of 2 different cell types. These cell types that we knew came from the embryonic notochord, that are big and very foam-like when you see them under the microscope. And then there were these other cells that people just called NP cells because we didn't really know what they were. And this was a question that we felt was really important even when we were doing tissue engineering, because we knew if those cells were there, these notochord cells, we formed way better tissue. So this is like intact 3-dimensional tissue we grew in a lab. And so these notochord cells we know do great things. And the question is, what's the relationship between these cells? And if we want to tissue engineer a disc, shouldn't we understand what cell type makes it in the first place? And so there were 2 theories at the time. One was that these notochord cells were actually stem cells. They gave rise to the nucleus pulposus. They were super important. And the other theory was that they were just remnants from the embryo. They formed the tissue during development, and then they died off and other cells grew in. And so some of the very first experiments from my lab sought to answer this, which of these models was actually true. And so we worked with a mouse model. And you might think that working in mice to study spines might be weird because yes, they do walk around on 4 paws, not on 2 legs like us. But it turns out that the biology, the structure of the spine is really consistent and conserved between humans and mice. And so we were able to take advantage of the mouse to do this. And one of the things that makes the mouse so exciting to work with is that we can modify their genes. So we have tricks and tools that let us go in there and change genes and add things and take things away. And it really lets us follow what's going on. And I was able to, when I started my lab, take advantage of some work that was done in the lab where I trained and take some of that with me, where they had found a way to genetically label cells of the notochord. And they were using these mice. So in here, all those mice are engineered to express fluorescent proteins. So these green cells that you can see are these specialized cells in the mouse embryo. You can follow them. They were studying what happened in really early development, but our question was, what would happen in the spine? Are those cells from that notochord important? Are they the stem cells of the spine? And so one of my first PhD students, Matt McCann, was able to use these animals. So in the images that I'm showing you, these are mouse embryos really early during development. So 5 days post-fertilization, at 12.5 days, where you can see limbs are starting to form and they have beautiful tails all the way through till full spine formation in the embryo. And we were able to ask, where do notochord cells go? And so in this scenario, they're traced in blue. They're genetically engineered so that all cells that come from this lineage express these marker proteins. And we were able to find that, yeah, during embryonic development, all the cells in the disc come from the notochord, which was really neat. But we were able to let those animals be born and grow to older age. And for the first time, we were able to show that all cells in the nucleus pulposus do come from this progenitor population. So they mature and they give rise to, they are the stem cells of the disc. And so this was a really neat discovery because it let us understand that this isn't really just a cartilage cell type. It's a unique cell type that comes from a specific place, and gave us these neat tools to now be able to isolate these cells. If we can see them, we can isolate them out. So here's a picture of a mouse tail where we've engineered the cells from this one lineage to express green fluorescence. And so we can actually dissect them out. And all these bright green glowing cells are these nucleus pulposus cells that we don't know anything about. And now we can use them if they're fluorescent, we can isolate them using all these techniques in the lab. We can study how they change over time to actually understand what these cells are and what makes them unique. And in a study that we did just recently, how do they change with age, and why is there so much disease that happens in the disc? So really neat tools, and the mouse is such a neat model for that because we can add genes and take away, we can track cells, we can knock out things, and they're really powerful tools in biology to understand what cells are or what's important in what a cell does. One of the other reasons that we like using mouse models is that they let us do really controlled experiments to ask specific questions. So I'm going to tell you a bit about this project that we did on the role of obesity. And you might be wondering, what does obesity have to do with spines, and what is going on here? So clinically, what we see with chronic conditions like back pain and degeneration of the joints of the spine is that there's all these associations. But anything that's associated, that a disease that is chronic and happens with old age, so many things are correlated, right? So with increased age, you have increased, the cells slow down, you have increased obesity, you might have osteoporosis, you might have all these other like confounding factors, right, that add up over time. And so we see that exactly with obesity, and there's this massive clinical association that doctors tell us, and they see it in the population between people who are obese and people with back pain. And the question is this a real association? So does obesity actually cause it, or is it just there at the same time because of age? Because obesity is really an interesting disease, because it's not just the extra weight that a high body mass has that can cause degeneration of joints. Obesity is actually a condition of chronic inflammation, that the high amount of adipose in your body actually triggers chronic inflammation throughout your entire body. So the question that we wanted to ask is, we see it in the clinic all the time, we see it in people. Is this does one cause the other? Does obesity cause disc degeneration? And does obesity cause back pain? And that's a really hard study to do in humans, because usually you can't track them their entire lives, and there's so many confounding things that are involved, but you can really control for this in animals. So I've thrown this up just to show you some of these kind of clever things that we can do in well-controlled animal experiments that let us ask really clinically relevant questions. So we can take mice that are young and healthy, so at 10 weeks of age, and in this study, we put the animals on either a normal diet, just like a standard lab diet, or a high-fat diet. So most of their calories came from fat. And we also put them on a Western diet. And Western being, high fat, high sugar, like your classic McDonald's fast food, Western living diet. And we said, what would this do? And we were able to follow these mice almost all the way to a year old and ask, as we're over time inducing obesity, what are we doing to the skeleton? So some really neat studies. And we could ask questions too about pain, as well as like the actual changes in the joint tissues that are associated with disease. So really intriguing. These are some neat X-ray images that show what these mice look like at the end of these diets. So hopefully in these pictures, the skeleton and any bony tissue is in white, fat is in yellow, and lean muscle mass is in red. And hopefully you can appreciate these mice got really big over time. So our diets were definitely able to induce obesity that was a pretty significant and really quick. And in fact, these animals became clinically obese after just 12 weeks on the diet. So it didn't take very much time. So we had this great model system where we knew, okay, over time, we have all these animals, they've been on this diet. What does this actually do? You might think to yourself, how do you measure pain in a mouse? Because when you guys go to the doctor, when we go to the doctor, to understand if you're in pain, you do pain questionnaires. And no matter how hard we try to train our animals, they're really bad at filling out questionnaires. So we work with some really clever collaborators who study behavior and have actually developed tools that let us ask, are mice in pain? And so I've thrown some of these slides up here just to show you that even though we can't communicate with the animals, we have different tests that we can run them through where we look at their locomotion, how much do they move, how do they react when stretched. So in an animal, even us, when we're in back pain, if we stretch the spine, you induce a pain reaction right away. And so we have these different tools that measure stretch pain or radiating pain, like you would in the clinic. We can actually do this on our animals that shows us if they're in pain. And what we were able to show was that over time, extended time, with these diets, animals showed both indicators of pain of both axial pain or back pain, as well as radiating pain. And not surprisingly, when we looked at these animals, we were able to show that obesity did indeed cause degeneration of the spine. So looking within the spines, looking within the tissues of the spine, we were able to see changes that are associated with disease, and looking even at the molecular level within the cells to see what genes are expressed and how are they different tissues from one group compared to the other. We were able to show many of these hallmarks of disease, so inflammation and breakdown of the tissues, that for the first time let us show that obesity does cause disc degeneration, at least in these animal models. Now, the last little story that I'll tell you, I've picked because it's a really neat one that comes back to the advice of just be curious. And so there's studies that are involved with the spine, but it's studying a disease that isn't just disc degeneration. It's a disease called diffuse idiopathic skeletal hyperostosis, which is a crazy mouthful. It's referred to as DISH, but it's really just well defined as ectopic bone formation of the spine. So your spine that normally has all these joints and is really flexible actually has all of this bone that forms, and it looks like dripping wax down the front of the spine. So this is actually an X-ray of a patient with DISH that has all this extra bone that forms along their spine, which obviously causes a lot of problems. This disease is actually the second most common form of arthritis, even though not a lot of people know about it. It affects over 25% of males over the age of 50. So crazy common. It has lots of symptoms like pain and stiffness. If these bony outgrowths get too big, it can stop you from swallowing. It can push on the organs and the tissues adjacent to it, causing all these problems. So it's a really important problem that we know very little about. In fact, it's been in the literature for 100 years, and we can't do anything about this disease. There's no drugs. There's nothing that can be done to slow or prevent this mineral that forms in your spine. Now, we knew nothing about this disease about 8 years ago when we accidentally started this work. So let me show you this video. It's a 3D X-ray of a mouse where anything in red is mineralized or bony tissue where it shouldn't be. So you can see all along the spine of this mouse, these big red areas, this really dense bone that's forming where the soft joints should actually be. So I've had a team of really talented trainees that have worked with me. And the great thing is this mouse I got a knock on the door one day from a researcher working in like cancer drug metabolism of all things, who was working with a knockout mice. And they had let the mice age longer than they normally do. And the mice were becoming paralyzed. And the vet did an autopsy, and they came down and said, these mice have crazy mineralization of the spine. And so they came to my door and they said, Dr. Seguin, you're a spine researcher. What do you know about this? And so we started studying these mice because it turns out that they show many properties of DISH. So our questions have been, is this a disease model? Could we use this to understand where things start, where they happen, and what controls the process? So these are more of these X-ray images of wild type mice and this genetic knockout mouse, showing that the spine starts calcifying over time, and it's progressive. It always starts in the neck and it spreads going downwards. And by 1 year of age, all of this bright white material detectable by X-ray is this ectopic mineral that's just formed in the spine. And so they're really big. You can see it with the naked eye. If you were to open up an animal or look at the spinal tissues of an animal, when we look by X-ray, there are these really big nodules of mineral that forms. And the neat thing is it's not actually bone. It's just chunky, chalky mineral that forms within the joints or the soft connective tissues of the spine where it really shouldn't be. And we can localize these using high resolution X-ray. And it's been a really neat model because we know where it's going to form, in what tissue. And that's let us do these really neat studies where we can isolate tissues from these animals and compare them to genetically normal mice. And do total discovery science. So we know when it's going to form, we know when it's going to start. And so we've done these neat experiments where we've asked, what are all of the genes expressed in these tissues at this moment in time? And then also, what are all the proteins that are there? And all of these tiny little dots on all these graphs are just proteins that are different between a wild type mouse and a knockout mouse. And hidden somewhere in here is the answer to the question of what causes this disease, and maybe some targets where we might be able to use drugs that could stop this process, to try to stop this from happening. So really neat work. And we worked in mice for quite a bit of a time, and we said, well, how do we know if this really is what's happening in humans, which is fundamental to why we're doing this research? And so I had a student who's now doing his PhD with me in a really neat combined physiotherapy PhD program. So at the end, he's going to have both of those degrees because his real interest is understanding and coming up with interventions for humans. And so we asked the question, well, is this mouse the same as humans? Because they show this weird bony stuff that forms, but it's not real bone. And so we said, well, geez, the problem with patients with these diseases is they don't get surgery. We can't access spine tissues. Everyone still needs their spine. Nobody wants to give it up. And so we developed this great collaboration with the anatomy teaching labs at the university, where people donate their bodies to research, to medical education. And so we were able to access spinal tissues from these donors. And we figured, well, generally speaking, these are people of advanced age. There's got to be at least one or two of them that might have this disease. And then we'd have access to tissues and we'd be able to compare. And it turns out it wasn't just one or two. It was great. Actually, half of the spines or half of the patients who had donated these tissues had evidence of DISH in them. So they met it. We had clinicians come in and look at the imaging and diagnose the spines for us. And this gave us this amazing opportunity to ask what happens in human tissues and does it look like our mouse model? So these are just some images of these human spines. So the vertebral bone with the intervertebral disc in the middle. And Dale, my student, was able to show that in these humans, you get these massive accumulations again. So here by X-ray, anything that's bright white is mineralized tissue. And within the soft connective tissues where the disc should be, instead, there's these big pockets of mineral. And we were able to show by looking at them using all these advanced X-ray imaging and basic histology, that these areas showed very similar patterns of ectopic mineral, very similar to our mouse model. And so while it might sound funny that you're excited that the mouse is like the human, the reason that we're so excited is that in the mouse model, we can look at really early stages of disease, look at mid stages of disease, and access tissues that in the humans we don't have access to. And that gives us the opportunity to apply, do all this beautiful cellular biology to try to understand what might be driving it. And having an animal model lets us test drugs and see if we can slow it or prevent it. And so it's a really neat tool that we hope is going to let us answer some important questions for a variety of different diseases. So that's the end of my talk. I throw this slide up all of the time because I think one of the best things about being a researcher is the amazing teams of people you get to work with. So here are some images of my lab over time. Students have contributed to this project from a bunch of different eras now, and acknowledging that they're the ones that every day get to come into the lab and work with the animals, carry out the experiments. And in addition to that, we work in a really neat environment at Western where basic researchers work with clinicians and our students move freely between the labs. And that to me has been really important. I work with, you know, lots of other researchers who have expertise that's different from mine, but lets us ask better questions when we work together as team. And so I like to acknowledge that kind of really wonderful team aspect to it, in addition to the funding agencies that have provided the resources for us to be able to complete this research.
We just have like a few questions that we wanted to ask. So I understand that your lab relies heavily on the mouse model to perform experiments, and that it's a good model to experiment with because the spine structure and like development is like super conserved, like in vertebrates such as humans and mice. And I was just wondering if there are any differences between mouse and human spines and how that would affect translation of your research to like a clinical setting, like where it would be like applied to humans.
Absolutely. And it's a really important question, right? There are many things that make us similar to mice, but many things that make us very different, right? And so acknowledging those differences is really important. So one of the most obvious one is the quadrupedal animal has a horizontal spine and we have a vertical spine, right? And so one of the most common questions I get all the time is the loading different? You know, when we walk around, our spines are loaded like this, and gravity is such an important part of it. And what is loading like on an animal spine, and how different is that? And it turns out that the mechanical load experienced by a quadrupedal animal is actually equivalent to or greater than the mechanical load, like in the axis of the spine, that we experience from gravity, because you can think about it like a roll of lifesavers. So if you stack them this way, you can remove your fingers. You don't need to hold them. Those lifesavers will stay lined up and stay horizontal. Now turn it this way. And if you don't push on either end of your little stack, all those lifesavers are going to fall out in the middle, right? So our bodies and the bodies of quadrupedal animals have adapted to put pressure on the spine using the ligaments and the musculature to keep the spine intact, even though it's positioned this way and not this way. So it turns out that's one of the things that makes it a good model system, surprisingly, even though it's a quadrupedal spine and not a bipedal spine. Some of the things that are different come back to the cell types. So it turns out that in rodents, those beautiful notochord cells that I told you were so important, they last longer into the animal's life. So you can still see some of those cells even in like an adult, whereas in humans, those cells disappear somewhere between the age of 10 and 15. And so disc degeneration in humans starts happening sooner than it does in some of these rodent models, because those cells mature at different rates and show so subtle differences. Obviously so in the disc, things are the same, but metabolism and body, and there's lots of really important differences that mean that any of those discoveries, right, have to be validated at multiple levels. But it's a pretty wonderful place to start.
Yeah, thank you so much. That was a really good analogy.
Moving on to the next question, I would really like to ask, how do you see your research impacting clinical practice and improving patient experience for those that suffer from back pain?
That's the goal. So, you know, the long-term goal of our program would be to contribute to that. So to contribute to understanding, you know, how things start and the biology that drives disease. And if we can start unraveling that crazy mystery of the biology that causes disease, we then know what to tweak and what to target in the context of disease modifying therapies. So, you know, our goal is to use this discovery platform that is basic research to be able to contribute to those understanding and to put that information out there, right? So to put all of this out there in the world so that if drug companies have compounds that target something that we've identified, if, you know, really clever young high school students or university students have a really great idea of how to use that basic information to take it one step further and develop a therapy, then that would be the goal, so that these big information, all these big experiments that we do can at some point come back to the clinic and to improving the health and the long-term mobility of patients.
Yeah, and I think it's so important, especially after your presentation and everything that we've learned so far. Like back pain is such a prevalent issue, and we need to, you know, have these like research labs and stuff to sort out that problem. So thank you so much for your answer. And also moving on to questions about just your life in general. What was your sort of educational pathway that led pathway, and how did that lead you to research about the intervertebral disc and the spine in general? Like, how did you come into this field?
Yeah. So when I came to university, I knew I wanted to study something about biology, and I didn't understand what was important. I remember sitting with like a guidance counselor in high school, and I'm like, this university has a program in immunology. What is that again? And this one has one in genetics. And what does it matter? Like when you first come, and things were different than some of the more general programs that we have now at universities. But as an undergraduate, I was able to start taking, you know, in second and third year different courses, and I found genetics mind-blowing and so intriguing, right? And all the way from little Punnett squares to understanding complex diseases and all the rest of it. And then in the current climate, be able to use genetics as such a fundamental tool to understand everything. So for me, it was just like kind of the puzzle of genetics I found really intriguing, and it was really a natural fit for me. And it's kind of, you know, I had such brilliant opportunities and accidentally walked through a few doors, to be really honest with you. So, you know, I was studying cartilage biology, and in the lab where I did my PhD, when I spoke with that researcher, I was initially coming to do a project on cartilage, on knee biology, on knee tissue engineering. And by the time I got there, she was like, oh, by the way, you'll be working on this new grant that I just got to study the disc. And I had to go figure out what it was. But it turns out it was just, you know, a really great opportunity to start working in a tissue that not a lot of people knew stuff about. And I always find that intriguing, right? Like walking into a really big question so that you're just kind of picking away and trying to understand. And then stem cell biology kind of was great too, right? And development, like understanding these basic processes. So for me, it's really been putting all those pieces back together again. So when in my own lab, I'm able to combine all of that in neat new ways, to be really honest, and something that I think students should really know, most of what drives what we do are the brilliant ideas of the students that get to train, that I get to work with, right? That do their degrees in my lab, because they'll come to me with these wonderful ideas, and we'll kind of clean things up together, and I'll say, yeah, go try it out, see if it works. And you know, while not everything works, which is really important to understand in science, when something works, it's really great. So, you know, for example, all the pain work was stuff that one of my students came to me and said, hey, I read this paper. There's this great researcher in Montreal who's trying to study back pain. Can we do it? I said, yeah, sure, try it out, see if it works. And now it's something that we do in so many of our models. So, you know, the great thing about science is you never really know what you're going to do when you wake up in the morning. Like you have some idea, but you know, you discover, you observe something, and it just kind of takes you in this weird way. And, you know, that's one of the reasons I wanted to tell you guys about the dish model, because that was literally somebody who knocked on my door and said, hey, you know something about spines? What's going on here? And by just asking questions and being willing, just being curious about things, it actually, you know, started this wonderful area of research about a disease we knew nothing about, but was so common. We're like, we know stuff, we can do stuff. Let's apply some of our tools to try to fix this problem. So just keeping things open and being willing to just ask questions and look at what's in front of you without too much of a preconceived idea is really what got me where I am. If I had to draw like a map of where I thought I would be, this wouldn't be it, but it's a pretty wonderful place to be.
Yeah, I think that's definitely really good advice, especially for people that like are in the position that you were when you were choosing your university degree, who are kind of unsure of what they want to go into, but like only certain about a certain direction. And we just have one last question. It can be like a short one, but could you briefly walk us through a day of working in your lab and talk a little bit about what experimental techniques that your lab regularly uses?
Oh, sure. So I'd love to tell you I'm actually in my lab most days, but unfortunately I'm in my office writing the grants or writing the papers about things. But our lab, so our lab is run by technical staff. We have one staff member who maintains all the animals within our animal care facility, and then students that are working on different projects. And so, a normal day in our lab will involve, may involve working directly with the animals to do those behavioral tests to look at pain. It may involve bringing them over to the imaging facilities to be x-rayed. Or it may involve working with the tissues at the end of the experiment, right? So growing cells from the mice. So often we'll do work with cells so that we can expand them. We'll isolate tissues so we can do specific experiments on the tissues. And a whole lot of teamwork, I think, would be the end of it. So, we try to create a neat environment in our lab where students work together, and there could be one day where everybody's helping out somebody. They all have their own projects, but they have common techniques like histology or gene expression analysis, looking at what's going on within the tissue so that they can help each other out along the way, and then everybody gets there faster.
Well,
thank you so much
for walking through your day in the life. It's always very interesting to learn about these different professions. Thank you so much, Dr. Seguin, for sharing your research with us. This presentation has been very enlightening, and we learned so much from you about the spine and embryological development, the applications of intervertebral discs, physiology, and about the relationship between obesity and back pain, which personally I didn't really know there was one. We also think this research is so exciting and holds a lot of promise for the world of research. And also just learning about how curiosity is vital in research and how that played a big role in your journey was very insightful and motivating for us as well as students. And in terms of YouthConnect, we still have so many more events planned, including more webinars and an upcoming conference in the summer. So please stay tuned, and you can all get updates about all of our initiatives by following our social medias in the description. If you are interested in Dr. Seguin's research or have any additional questions, or just want to tell her how cool her research is, you can contact her through email, which is also in the description. Thank you so much once again, Dr. Seguin, and see everybody in the next webinar. Bye.